Assalamu alaikum. So in this lecture, what we will do is we're going to cover the metabolism of iron, and that includes absorption, distribution, excretion, if there's any, and disorders related to metabolism of iron. There are several resources that you can rely on. Of course, first is the lecture itself, as I said. The second is this really nice review written by these two individuals, Latonde, Dada, that's an easy one, and this one right here. So 5 dinars for whomever can say the name. So anyhow, so this is an updated review, and I have found it to be an excellent one. You can refer to it. You can also refer to Lippincott's biochemistry textbook, the 7th edition, the 7th edition. So you can, as I said, you can go to the index and look up different terms related to this lecture. Now, there are other resources that you can refer to if you wish. The medical biochemistry page, as I said, it's an excellent one, as well as these two review articles. So let's talk about iron first. Now, in the body, iron exists in two oxidation states. You have the ferrous form and you have the ferric form. An important point about the ferric form is that it is highly insoluble. So keep that in mind because it's important when it comes to iron absorption and transport. Now, iron is important for the function of many proteins, specifically enzymes, and more specifically cytochrome P450 enzymes, where it functions as a prosthetic group. That is, it binds covalently to the enzyme, and it's important for enzyme function. Iron is important for metabolism as well as oxygen transport, and of course, hemoglobin, of course. So, however, now iron can be toxic, especially if it exists in high concentrations or levels in the body, because it forms free radicals and it can cause tissue damage. So as a result, hardly ever you can find iron in the free form. Rather, it's mainly found bound to other proteins. So iron is precious in our system. Normal individuals have about 3 to 4 grams of iron. The amount of iron that is absorbed daily is about 1 to 2 milligrams daily. So compare this to how much iron we have in our system. That's very little iron is absorbed. And the amount of iron loss is also very little, 1 to 2 milligrams daily. Okay. So of course, women would lose more iron than men due to the menstrual period. Now, most of the iron in our system exists in erythrocytes. About 2,500 milligrams of iron exists in those cells. We have cell renewal. A lot of cells die daily, okay, and releasing heme and releasing iron. Now, this iron is sequestered very efficiently by cells of the radiculoendothelial system, specifically the macrophages. And some of the released iron would be immediately bound to transferrin in plasma. So we have about 4 milligrams of iron bound to transferrin protein, which the released iron can be recycled by bone marrow cells to produce more erythrocytes and more hemoglobin. Now, some iron can also be stored in hepatocytes as well as Kupffer cells. About 1,500 milligrams of iron would be stored. Now, also about 300 milligrams of iron would be used to produce myoglobin as well as enzymes. So let's talk about the life cycle of iron in our system. We're starting with absorption. So iron in our system exists either, as I said, in the ferrous state or the ferric state. So this is the ferric state, this is the ferrous state. Now, in alkaline conditions or neutral conditions, iron would exist in the ferric state. And in acidic conditions, like in stomach, it exists in the ferrous state. So, in order for iron to be absorbed, it has to be in the ferrous state. So once it reaches the small intestine, it gets converted from the ferrous state to the ferric state. Now this ferric iron must be converted to ferrous again. And this is done by an enzyme that is membrane bound known as ferroreductase. Now, in order for iron to be transported into enterocytes, it has to be reduced first from the ferric to the ferrous state. And this is done by an enzyme, a ferric reductase or a ferroreductase enzyme known as DCyTB, duodenal cytochrome B. This enzyme exists on the cell surface, and it is associated with the transporter, which is known as the divalent metal transporter 1. This is an important transporter that exists not only on the cell surface of enterocytes but other cells as well. Now, it is an enzyme that is not specific for iron. It can also transport other metals like zinc, copper, cobalt, magnesium, and so on. Now, the thing is, where does the ferric reductase enzyme get an electron from to donate to iron? And it comes from vitamin C. So it extracts an electron from vitamin C in the cytosol. And this electron is then, it can move outward or to the outward portion of the enzyme, and iron can then be reduced. And this is why vitamin C is considered an important supplement for iron metabolism or absorption. Another important source of iron is heme itself. And of course, heme comes mainly from eating meat. So heme can be taken up by enterocytes through a transporter or a carrier protein known as heme carrier protein 1. So the whole heme goes into cells, and then iron is extracted by an enzyme known as heme oxygenase 1. Now this enzyme right here does not exist in enterocytes only, but it also exists in other cells like macrophages, for example, and it has the same function, that is extracting iron from heme molecule. So once iron exists in these cells, it has one of two fates. It can be stored, that's fate number 1, by binding to a protein known as ferritin. Now ferritin can bind to about 4,500 iron ions in the ferric state. Now the thing is, if iron is stored in these cells, in the intestinal cells, now it would be lost because some of these cells would be shed, they would be sloughed off, okay, and iron would be lost. But in order to take advantage of iron, it can be transported out of intestinal cells via a transporter known as ferroportin, or a channel known as ferroportin. Now this ferroportin is not specific for intestinal cells, but it can be found in other cells such as macrophages. So there are conditions that result in abnormally low level of iron in the body, and these conditions are related to the intestine itself. Now the two conditions are iron malabsorption, so there is a problem in absorbing iron, and the other is intestinal hemorrhage. Now both of these disorders cause low level of iron in the body. Now in terms of iron malabsorption, it can result from gastrectomy, celiac disease, which results from death of enterocytes, Crohn's disease, and Helicobacter pylori. Now, Helicobacter pylori causes ulcers, but it's a different mechanism that it uses to cause iron malabsorption. Now, intestinal hemorrhage is basically abnormal bleeding that leads to inability of cells to absorb iron, and there is excessive loss of iron. And intestinal hemorrhage can result from gastric cancer, ulcers, whether stomach ulcers or duodenal ulcers, inflammatory bowel diseases, and it can result as well from a parasite, the hookworm. Now, these are some images of the hookworm. So this is in the gastrointestinal system. So you can see this worm. You can see it hiding in here as well. That's how it looks like. And here it exists right here in the bottom of the foot. Quite amazing, huh? Now, once it's outside the cell, it has to be oxidized back to the ferric form. Once it is in the ferric, and it can be oxidized by a protein, also membrane bound, known as Ferroxidase. Now, a similar enzyme can be found in the plasma protein ceruloplasmin. So ceruloplasmin has an enzymatic activity whereby it can oxidize iron from the ferrous to the ferric state. Once it is in the ferric state, it can then bind to the protein transferrin, the iron transporter. So transferrin is an iron binding protein. So it binds iron covalently, making transferrin a hollow protein. Without iron, it becomes an apoprotein, an apotransferrin. So it binds iron in the ferric state, not the ferrous state, and most of the transferrin in our system is actually not bound to iron. Okay. Now, each transferrin molecule has 2 iron binding sites. Most of them, as I said, about half of them are not bound to iron whatsoever. About 4 out of 9 of these transferrin proteins are bound to only 1 iron at one site, and 1/9 of the transferrin proteins are saturated with iron. This means that if you do it's math, that 1/3 of iron molecules of the binding sites would be bound to iron in our system. Now, in cases where there is high level of iron in the system, all of the iron binding sites would be saturated, and there would be increased levels of iron known as non-transferrin bound iron. Cells in need of iron display a receptor on the cell surface, and it is known as transferrin receptor, specifically transferrin receptor 1. So this transferrin receptor has the ability to bind to 2 transferrin molecule, or diferric transferrin. Now, the affinity of the receptor to the diferric transferrin is much higher than transferrin that carries just 1 iron atom, or the monoferric transferrin. And the monoferric transferrin has higher affinity than binding to transferrin than the apotransferrin. So once the transferrin binds to the receptor, this activates receptor-mediated endocytosis. So you have this invagination, you have this formation of a vesicle that transforms into an endosomal vesicle. Now, early endosomal vesicle. Now, this early endosomal vesicle would transform into a late endosomal vesicle. Now the difference is that the late endosomal vesicle is more acidic than the early endosomal vesicle. Now the low pH environment or the acidic environment inside late endosomes would allow for iron to be released from the transferrin receptor. Now note something, that in the vesicle itself you also have the divalent metal transporter, the same one that we talked about in enterocytes. So what happens is that once iron is released from transferrin, and note that the transferrin receptor is still bound to the transferrin itself. So once iron is released, it gets reduced by a reductase, and this reductase is called STEAP3. And then iron can be transported out of late endosomal vesicles into the cytosol, and it can be stored into ferritin. What happens then is that the receptor and transferrin, the complex, is recycled back into the plasma membrane in order to bind another transferrin molecule or ferric transferrin molecule. So let's talk about regulation of different proteins involved in the transport of iron. So Hepcidin is the main iron sensor and regulator in our system. It's a peptide hormone. It's composed of 25 amino acids secreted by liver. And when iron levels increase, and in cases of inflammation, hepcidin secretion increases, and that would reduce iron absorption. On the other hand, when iron levels are low, the release of hepcidin is decreased. Okay? So, what hepcidin does is that it reduces specifically the amount of ferroportin that exists in the basolateral side of intestinal cells, reducing the release of iron into our system. So iron would stay in intestinal cells, and these intestinal cells would be sloughed off, they would die, they would shed, and iron would be lost. So how does hepcidin do that? Well, if there are high levels of iron, that would increase the release of hepcidin. Now hepcidin can bind to ferroportin, and it induces the internalization of ferroportin inside cells into lysosomes, and ferroportin would then be degraded in lysosomes. Now the thing about hepcidin also is that it can also regulate the amount of divalent metal transporter. Okay, so this is the one that exists in the apical portion of intestinal cells, and is responsible for absorbing iron into intestinal cells. So hepcidin inhibits the production of divalent metal transporter, so little iron would be absorbed by intestinal cells. Now, due to the importance of hepcidin in regulating the level of iron in the body, it has to be highly regulated as well. Hepcidin itself has to be highly regulated. And it can be regulated via 4 mechanisms as shown in this slide. Now each mechanism is written in a certain color. So let's start with the first mechanism right here. Now on the cell surface, you have the transferrin receptor 1 right here. And it is associated with another protein known as HFE. Now this protein right here, sort of like it prepares transferrin receptor 1 to bind to transferrin, to ferrotransferrin, the transferrin that is bound to iron. Now, once this ferrotransferrin binds to transferrin receptor 1, HFE protein dissociates from transferrin receptor 1, and it goes and it binds to another protein. Now, this protein is transferrin receptor as well, but it's transferrin receptor 2, and it has another function different than the function of transferrin receptor 1. Its function is to send the signal inside the cell telling it that enough iron into the body or inside the cell. So what it does, what transferrin receptor 2 does, is that it sends a signal to induce the expression of hepcidin, and hepcidin would then block iron absorption, iron uptake into cells, and so on. So it's sort of like it balances how much iron should be in the blood and how much iron should be inside cells. So this is the first mechanism. Now the second mechanism is mediated by a protein known as bone morphogenic protein 6. Now this protein right here binds to a receptor, and its receptor is BMPR. So and what it does is that it activates a signal transduction pathway mediated by a transcriptional regulatory protein known as SMOD. What SMOD does is that it induces the expression of hepcidin. Now the receptor itself, the BMPR, is associated with a protein known as hemojuvelin. Now hemojuvelin is important for the function of this receptor. Now this hemojuvelin itself is regulated by a protease known as TMPRSS6. Now this protease right here is important, by the way, these days, because it activates the spike proteins of the coronavirus, allowing the coronavirus to get into cells. So here's a link to the coronavirus. So we have this signal transduction pathway induced by this sensor, the BMP6. There is another signal transduction pathway that is activated or induced by interleukin 6, an inflammatory cytokine. And what it does is that it binds to its receptor, it induces another transcriptional factor known as STAT3, and STAT3 induces the expression of hepcidin. Now the reason why you want to stop iron absorption and uptake into cells during inflammation is that inflammation indicates that there is an infection, and pathogens might feed on iron. They flourish if there is high level of iron. So as a protection mechanism, there would be reduction of iron absorption and uptake into cells by hepcidin in order to protect the body. Now a fourth mechanism is hormonally regulated. And what happens is that in cases of hypoxia or anemia, this is basically sensed by kidney cells. So you have the induction of a protein, an important regulatory protein known as hypoxia-induced factor. It induces the expression of the hormone erythropoietin from kidney cells. Now erythropoietin then functions on erythroblast to induce the expression of another hormone or factor known as erythroferron, and erythroferron would inhibit the production of hepcidin in order to compensate for the low iron level in the body. So let's talk very briefly about post-transcriptional regulation of iron regulatory proteins. So iron regulatory proteins have something in common. and that is they all have in their messenger RNA molecules iron response elements, and these elements are the sites whereby iron regulatory protein can bind to. So you have and the position of the iron response elements is different among the different messenger RNA molecules. So for example, for the ferritin messenger RNA, the iron response element it exists at the 5' untranslated region. On the other hand, for the transferrin receptor, the iron response element exists in the 3' untranslated region. So why is that important? Because when the protein, the iron regulatory protein, binds to the iron response element at the 5' untranslated region, it prevents translation from this messenger RNA. And if it binds to the iron response element at the 3' untranslated region, the messenger RNA becomes stable. So if there are low levels of iron in the system, iron regulatory proteins would bind to the elements. In this case, there would be low production of ferritin, and it makes sense, right? Because there is no iron, so there is no need to make ferritin, because there is no need to store iron. So translation of this messenger RNA would be blocked. At the same time, we want to increase the production of transferrin and divalent metal transporter 1 by stabilizing the messenger RNA, okay, because there is low level of iron. Now, if there is high level of iron in the system, iron would bind to the iron regulatory protein, releasing it from the element at the 5' untranslated region and the 3' untranslated region. So if it is released from this messenger RNA right here, translation resumes, increasing the level of ferritin protein, storing excess iron in the system. At the same time, for these messenger RNA molecules that have the element at the 3' untranslated region, releasing the iron regulatory protein would lead to destabilization of the messenger RNA, and the messenger RNA would be degraded, reducing the amount of transferrin and divalent metal transporter 1, for example, because there is no need to absorb and transport these proteins. Let's end this lecture by talking about iron-related diseases, and there are two diseases that we want to cover. The first is hereditary hemochromatosis, and it's a group of disorders. It's not just one. And we have the iron-deficiency anemia. So the hereditary hemochromatosis, basically, it's a group of disorders related to iron metabolism, and it is this group or these disorders result in excessive iron absorption, saturation into the body, as well as deposition into what is known as hemosiderin in the tissues. And hemosiderin, we'll talk about it in a few slides, is toxic to tissues. Of course, hereditary hemochromatosis is more common in males rather than females, and I guess the reason is obvious, due to menstruation. Now, there are different types of or different classes of hereditary hemochromatosis. The main one is known as type 1 or primary hemochromatosis, hereditary hemochromatosis, and it's related to mutation in the HFE protein that I talked about before. But there are other genes, 4 other proteins that have been found to be associated with hereditary hemochromatosis as well. So the classes of hereditary hemochromatosis disorders are type 1, the most common one, and it's HFE dependent. Now we also have type 2, and there are two types. We have type 2A and type 2B. Now type 2A is related to mutations in the hemojuvelin protein that associates with the BMP6 receptor. And type 2B is associated with mutations in the hepcidin gene. Type 3 is related to mutations in transferrin receptor 2, and type 4 is associated with ferroportin protein. Now, all of them are autosomal recessive disorders except for the ferroportin dependent type, which is autosomal, which is inherited following the autosomal dominant pattern. So normal individuals have limited quantities of iron in the system, but people with excessive stores of iron, they can have up to 50 grams of iron in their cells. So this is a lot of iron, and this iron would accumulate, producing what is known as hemosiderin. So hemosiderin are basically water insoluble deposits in tissues. So here you have a tissue section with hemosiderin stained in brown. So that's a lot of iron deposits. Now the thing about hemosiderin and these iron deposits is that they can damage tissues. So they can damage liver, resulting in hepatic fibrosis, the pancreas, diabetes, joints, skin, heart, resulting in cardiomyopathy, and so on. So, hemochromatosis conditions all have something in common. There is high level of iron in the system. So remember that I talked about transferrin receptor 1 and transferrin receptor 2 having transferrin receptor 1 bound to HFE protein. That's in case there is little iron in the system, so transferrin cannot bind to transferrin receptor 1. When there is high level of iron in the system, transferrin would be saturated. It binds to transferrin receptor 1, releasing HFE so that now it can bind to transferrin receptor 2, inducing signal transduction, releasing hepcidin, or increasing the production of hepcidin. Now what happens in some cases of hemochromatosis is that HFE itself is mutated. So there would be, it would be unable to bind to transferrin receptor 1, and as a result there would be continuous entry of iron into the system, and there is little production of hepcidin as a result of that. That results in increasing intracellular iron in cells. Okay, so remember these figures. So if there is inability of hemochromatosis to bind to transferrin receptor 1 or 2, there would be increased intake or uptake of iron into the system. Now, type 2 hereditary hemochromatosis is also known as juvenile hemochromatosis. The reason is that symptoms appear at a young age. Now, there are two types of juvenile hemochromatosis, type 2A and type 2B. Now, type 2B is associated with mutations in the hepcidin gene. Type 2A is associated with mutations in the hemojuvelin protein. Now, this is the protein that I said before. It associates with BMP6 receptor, and it's important for its function, and this receptor induces the expression of hepcidin. So if there is mutation in the hemojuvelin protein, then there is no induction of expression of hepcidin. Well, the second condition that we want to talk about is anemia, iron deficiency anemia, which is basically it's characterized by a low number of mature erythrocytes in the circulation. That would result in lowering oxygen carrying capacity in the blood, resulting in tissue hypoxia. Now, clinical symptoms include fatigue, weakness, increased cardiac output, increased morbidity and mortality as well. And anemia is a very common disease worldwide, especially in poor countries. Now, anemia can result from deficiency of vitamin B12 or deficiency of folate. Now, both of these cofactors, both of these vitamins are important for the production of nucleotides. So folate is important for production of nucleotides, and vitamin B12 is important for regenerating folate during this pathway. Now what happens is that if there is deficiency of folate, that would result in the production of what is known as megaloblasts, large cells. These result from inability to synthesize DNA and inability of cells to divide. So you would have the production of these large cells. Now, what's really important as well is that I said that anemia is common in poor countries, but it's also common in more industrialized nations as well, such as the US and Europe. And it can result from chronic inflammation. The reason is that if there is chronic inflammation, that would result in overproduction of cytokines, that would result in production of hepcidin, large quantities of hepcidin by hepatocytes, that would result in downregulation of ferroportin expression in cells, such as intestinal cells and macrophages. That would result in inability or low amount of iron in the system, and that would eventually result in lack of production of hemoglobin. Eventually, you will have anemia. Now, there are other consequences of chronic inflammation that would include, in addition to increasing production of hepcidin, you can also have inhibition of erythropoietin release from the kidneys. That would result in decreased erythropoietic stimulation and production of red blood cells. These inflammatory cytokines can also inhibit erythroid proliferation as well. They can also augment hemophagocytosis and inability to release iron from the system. Okay, so they're all connected to each other.